Welcome back to my course, that is aspects of biochemical engineering. Now, in the last couple of lectures, I try to concentrate on the microbiology, which is involved in the biochemical process, and I told you that microbiology or microorganisms that plays very vital role as far as biochemical processes are concerned. Now, now today we will be discussing another very interesting topic, what you call fundamentals of biochemistry. Now, naturally, why, how the different bioproducts formation take place with the help of my organism, that we can find out if we go through this biochemistry of this living cells. We can understand the process very nicely. So, the first I want to tell you that what you mean by biochemistry. Biochemistry is the study of the structure and composition. You see, this is structure, composition and chemical reaction of substances in living system. That different components present in the biological system, how we take part in the reactions. That is what biochemistry deals with. So, it basically is a combination of biology and chemistry. That is the biochemistry that we have. Now, now question comes, what are the different biomolecules present in this biochemical process? That is very important, because these molecules are mostly either carbohydrate or proteins or lipids or nucleic acid. These are the different major biochemicals, that biomolecules present in the bioprocesses. Now, what do you mean by carbohydrate? Carbohydrate is considered as a source of carbon, which is, I told you, this is used for three different purposes. One is for the cell mass formation, and there is a source of energy. Also, it is used for the product formation, where the product is other than the cell mass. The protein is mostly used for the building up of the cells, and lipids also used as a source of energy. And if you look at the nucleic acid, this is mostly taking part for the formation of DNA and RNA, which is the genetic material of the living system. Now this is carbohydrate. If you see it very minutely, you will find it is usually ended with O's. That, as for example, that glucose, fructose, different type of the nomenclature is usually ended by O's. Now, it comprises of three major components. One is carbon, then hydrogen and oxygen. And as I pointed out, this is considered as a major source of energy, and it can be available in two different forms. One is called monomer, and there is polymers. Now, monomers, when it's kind of monosaccharides. Now, if you consider sugar is not a monomer, it is a dimer. Actually, it is the combination of glucose and fructose. Now, polymer is a starch, cellulose is a polymer of glucose. So, it is a polymer. So, examples, the same examples are giving here, the glucose, fructose, galactose. Galactose also dimer, that glucose and galactose lactose is a dimer, is a combination of glucose and galactose, and then we have sugar, starch, which is available, potato, pasta, etc. Now, if you look at the protein molecule, this is the most important, because it is used as a nitrogen source, and it comprises of carbon, hydrogen and oxygen and nitrogen. It controls the rate of chemical reaction through the enzymes. The enzymes are the basic that biomolecules that is mostly take part in the reaction, and enzymes, they are considered as the biocatalyst. And role of a catalyst, as we know, that it take part in the reaction. After the reaction is over, it remains unaltered. Purpose of the catalyst is to lower the activation energy, so that your rate of reaction increases. So, this is available in bone and muscle and transport things out of cells. And then it can be monomer. If it is monomer, we call it amino acids. Now, as you know, we have 20 different amino acids, and and this proteins, it is basically it is a polymer of amino acids, different amino acids. So, it basically it available in two different forms. One is called peptides, and there is protein. If the peptides is there, it contains 2 to 50 amino acids. Is more than 50 amino acids, we call it proteins. Now, it is available in meat, then poultry, eggs, beans, soybean, nut, peanuts, butter, and the enzymes. So, these are the different sources where these enzymes, these enzyme proteins are present. Then, as I told you, nucleic acid, which contains special thing it has, that is the phosphorus. So, it contains carbon, hydrogen, oxygen, nitrogen, phosphorus. It stored and transmit the genetic information, which is very important. That genes, as you know, any genes that code a particular proteins. So, and this protein, that enzymes are basically proteins, and this is very much essential for carrying out the biochemical activities. The monomers is the nucleotide monomers, and polymers is the polymer of nucleic acid. As for example, we have DNA deoxyribonucleic acid and RNA ribonucleic acid. So, this is how it looks. And then we have another molecules. I told you this is lipids. It is no true polymer. There is no true polymer here. So, we have this contains primarily carbon and hydrogen. Also, it contains oxygen. It is stored as energy, I told you, and make up of biological membrane and waterproof covering. That it is hydrophobic in nature because it has the water repelling properties. They are made up of 3 fatty acids and 1 glycerol, and the fats examples are fats, oils, wax, and the membrane. These are the different examples that we have. Now, next is let us go to this biomolecules in the details. Now, if you look at the carbohydrate, now as I told you that it is a source of energy, because it is the key source of energy of the living organisms. As for example, that we take glucose in the day-to-day life, and glucose we consider as a source of energy. It passes through the endothermic pathway, then TCA cycle, and ultimately it converted to carbon dioxide and water. Now, here I want to point out that this general formula of carbohydrate, as I shown you, this is CH2O. This is CX and Y. So, this is called carbo and this is called hydro. This is how. Now, if I consider glucose, C6H12O6. Now, how you can write it? C6H2O6. So, this is carbohydrate. Now, if this is about the glucose. Now, if you consider that other molecules, as for example sugar, how you can write? It is C12H22O11. So, this is C12 and H2O, this is 11. This is how we can write. So, this is carbo, this is hydrate. This is why it calls the carbohydrate. Carbohydrates are synthesized by photosynthesis process using sunlight, because this is the normal process that is going on in the living system, and schematic representation of the reaction is given here. You see that carbon dioxide and water in presence of light, it produces the carbohydrate, this and oxygen. This is how it, and the two functional groups are present here. One is called keto, that this is the ketone group, and another is the aldehyde groups. This is the two things that we have. According to the stereochemistry, two stereoisomers of carbohydrates are present, both D form and L form, and usually most of the natural sugar we have only the D forms. The D isomers are present there. Now, carbohydrate may be available in different forms. One is monosaccharide. Monosaccharide, I explained that it comprises of one unit, that is 6-H12O6, that one unit you have, and fructose also same as glucose, and galactose also this one monomer unit is there. Now, if you consider dimer, we have sucrose, lactose, and maltose. What is sucrose? It is the dimer of glucose and fructose, and lactose is the glucose and galactose, and maltose is a glucose and glucose. They form alpha 1,4 linkage to form the maltose molecule. Now, oligosaccharides, basically those are the polymer of the 2 to 10 sugar that they together is forms. One is raffinose, another is chartreuse, and then there is another polysaccharide that is we have which has the 10 or more sugar molecules are present. Those are those are present, that is one is starch, then glycogen, and the cellulose molecule. Now, that starch, both are polymer of glucose unit, and we know that cellulose is not digestible in the human system, but starch and glycogen they are digestible to the human system, and only the difference is that here we have alpha 1,4 or beta 1 or alpha 1,6 linkage. Now, in the cellulose we have beta 1,4 linkage only. So, this is the basic difference that we have. Then if you look at the protein molecule it is the high molecular polymer of amino acids, that if you look at this is the polymer of. So, anyhow, that protein is basically it is a high molecular weight polymer of amino acids, that and amino acid basically they have the polymer with that CONH bond. This is so it is a combination of ammonia and the carboxylic acid that is they form this CONH2. I can explain like this. Commonly found in this is commonly found all the enzymes that we have, and acts as a biocatalyst in the several biochemical reaction. And as I pointed out that if the polypeptides what is the polypeptides where we have less than 50 amino acids are there. If more than 50 amino acids there, we consider as a protein. So, condensation of polypeptides, if we have the combination of several peptides molecules, then we call it protein molecule. We have several proteins, we have collagen protein, we have proteins, a lot of protein molecules we have. Then another we have the prosthetic groups. In the organic, it may be organic, inorganic component other than the amino acids in the chains they can produce. The pictorially that protein molecule can be represented like this. It is the primary, that if you look at that primary, that structure is like this, that it is a polymer of amino acids, and then it has secondary protein structure occurs that the linkage with they have the hydrogen bond, and hydrogen bond is in between the carboxylic and amino acid with the R group of the amino acid. They form the hydrogen bond, and they have this alpha helix, they have splitted sheet like this. Now, in case of tertiary structure, it is a combination of both the things, alpha helix and plated sheet, their combination they form the tertiary, and quaternary structure is a protein consists of more than one amino acid chain. So, this is gives you a brief idea that how the protein molecule looks. Now, next come to the lipid molecule, and we, as you know, lipid is the major source of energy. If you look at the energy content in the lipid is much higher as compared to the carbohydrate molecules. Now, fats or lipids are formed through the esterification of long chain fatty acid with the glycerol. So, they form the esters. We know the fatty acid. What is fatty acid? Fatty acid, basically this is the organic acid. What is the organic acid? It is comprised of carbon, hydrogen, oxygen, that and with the carboxylic acid groups, and when they combine with alcohol, they form the esters. And the fatty acid, the fats or lipids are basically the esters of that fatty acids and the glycerol. Now, general structure of this is given here. This is the general structure of fatty acid is given here, that CH3, CH2, COOHN. So, it may be bigger, we have bigger fat molecules, we have smaller fat molecules that we have. Now, fatty acid may be two types, one is saturated and unsaturated. What do you mean by saturated fatty acid? Saturated fatty acid means that all the bonds are single bond, there is no double bond present in this. Now, if there is any double bond present in the fats or the fatty acids, we call it unsaturated. Now, why we are interested particularly in this connection, I want to tell you particularly biodiesel production process, we usually prefer the unsaturated fatty acid, because why? By unsaturated, if unsaturation, if the fat is unsaturated, the oil should remain in the liquid phase. Otherwise, if it is saturated, it will, in the normal temperature, it may solidify. So, it is very difficult to use as a fuel. So, so here the saturated fatty acid, no double bond, and unsaturated at least one. Usually there should not be any double bond, maybe mostly the single bonds are present. The steroids are also lipid, which examples are the cholesterol and naturally occurring hormones. Now, this it looks like this. This is I told you, this hydrocarbon molecules. We can increase the change. This CA 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 13, 14, 15, 16, 17. So, 17 carbons, and then CO, and so this is the ester bonding that we have, how it forms this esters. So, no, this is we have the carboxylic acid group. This is the fatty acid, and this is also fatty acid, and with the help of this alcohol, they form the esters. That this is how the oleic acid is the organic fatty acid, and this is stearic acid, also fatty acid. Now, nucleic acid, which is a very important as far living cells is concerned, because we know that deoxyribonucleic acid, this is largely available in the living system. This is the deoxyribonucleic acid and ribonucleic acid that we have, and they carry out the genetic code as well as responsible for protein synthesis. So, this is very important, and it has pentose, ribose, and deoxyribose. They are nothing but pentose. This is also kind of carbohydrate, are present in RNA and DNA respectively. The nucleotide is majorly built up of three different molecules. One is phosphoric acid, then pentose, then nitrogenous base. Now, what is called nitrogenous base? Here is the nitrogenous base. In case of RNA, and this is RNA, this is called DNA. Now, in RNA you have cytosine, guanine, and adenine, but the special nitrogen base is the uracil. But if you look at the DNA, we have cytosine, guanine, adenine, but we have specially present, that is the thymine. So, this is the specially present, this is how the DNA, RNA, they differ from each other. So, nitrogenous base includes the adenine and guanine, all these called as nitrogenous base, and this is called adenine and guanine we call purines, and thymine, cytosine, and uracil we call pyrimidine. Now let me go to the biological system. In the biological system, two things simultaneously take place. One is called anabolism, another we call catabolism. Now, what is called anabolism? Anabolism is the complex molecules are formed for as a building blocks, because when some formation take place, as for example protein formation take place, or some molecule formation take place, that we call it anabolism. When degradation of the molecules take place, as glucose, when we consider as the source of energy, glucose finally converted to carbon dioxide and water, we get the energy, that is called catabolism. So, the anabolism means formation of the body cells, and catabolism is the degradation of the organic molecules. So, that large molecules are broken down into the bonding bricks. Now, this is cells. We have micromolecules, macromolecules like the protein, then polysaccharide, then lipid and nucleic acid, and this form this different compound precursor, amino acid, sugar, fatty acid, and nitrogenous base. So, when this amino acid forms protein is nothing but polymer of amino acid, then this is called anabolism. When sugar form polysaccharides, then we call it that the anabolism. But other way, that if carbohydrate, fat and protein molecule is degraded to that carbon dioxide, water and ammonia, we call it catabolism process. I hope, and in process we produce some energy molecules. These energy molecules are ATP, NADH, NADP and FADH2. This is used as a currency of energy, because as per our body requirement, we use this energy for our day-to-day use. Now, major metabolic pathways are like this, that first we have protein molecules. When it degraded nitrogen pool, then it forms the ammonia. This is the ammonia that we have. Ammonia is formed, then the tissue protein that also it forms, and then carbohydrate. This carbohydrate basically the glucose, fructose and galactose, they take part glucose 6-phosphate, then it ultimately through the process of glycolytic pathway it produces the pyruvic acid, and then also this pyruvic acid when passed through the TCA cycle or citric acid cycle, and it produces carbon dioxide and water, then we get lot of energy. We produce the ATP, different electron transport chain. So, this is carbon, oxygen is required. So, two major products we have, carbon dioxide and water, that is and then in case of fat and lipids, we have fatty acids and lipogenesis, that both is taking place in this particular system. Now, metabolites are the intermediates or products of metabolism, because that I told you that in a chain we have several product formation take place, and individual molecules we call it metabolites, and it is broadly divided into two different types. One is called that primary metabolites, another we call the secondary metabolites. Now, what is called primary metabolites, like amino acid, vitamin, nucleic acid, polysaccharide, ethanol and lactic acid, and secondary metabolites are antibiotics, pigment, toxin, alkaloids and steroid and polymeric substance like gum and rubber. This is usually formed, this all usually formed during the stationary phase, and as I told you that stationary phase we consider as a starvation phase. Now, common that glucose metabolism is like this: glucose pass through the embryonic pathway, it produces the pyruvic acid, and through the both the aerobes and anaerobes they follow this pathway, and through the aerobes it passes through the TCA cycle, Krebs or citric acid. Meaning is more or less the same, that so then it passes through the respiratory chain or oxidative phosphorylation. The anaerobic system, it is a fermentation, ethanol and acetic acid and lactic acid it forms. Now, this is the glycolytic pathway, how the glucose molecules is converted to pyruvic acid. So, this is the how the number of this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, 10 different steps are involved before it produces. So, 1 mole of glucose can produce 2 moles of pyruvic acid. Then we have that aerobic glucose metabolism pathway. There we can produce some kind of it passes through the TCA cycle, and you can see the different molecule like citric acid, that kind of citric acid formation. When you take, we try to inhibit the subsequent steps by putting the inhibitor for aconitase enzyme and isocitrate dehydrogenase, we get the citric acid. We can synthesize different amino acids by using this process. Then we have the microbial metabolic pathway for industrial important products, like we have glutamate. You can see the glutamic largely used. Then we have other different amino acids are here, and that is how it forms by using this metabolic pathway. Then other industrial importance products are, as for example, different type of antibiotics are there. One antibiotic is the novobiocin. This is kind of antibiotics. Actinomycin, that is another antibiotic. So different type of antibiotics, how it forms, it has been shown here. Then other anaerobic fermentation process, we get different type of various chemicals. We can get like propionic acid, isopropanol, butyric acid, butanol, ethanol, which nowadays plays very important role because as a source of energy, and particularly lactic acid is largely used for the food preservation purpose. So it is used in the food and pharmaceutical industries, and polylactic acid is used. Polylactic acid used in the therapeutic purpose. Now, this is that microbial metabolic pathway for industrial importance. We have different species. We have lactobacillus, we have Saccharomyces, we have Clostridia. These are the very important species we have. As for example, lactobacillus is largely that when you take part, it produces the lactic acid, and which is used for the cheese production, sauerkraut production, yogurt and pickles, largely used. And Saccharomyces cerevisiae is used for wine, beer and ethanol. Ethanol acts as a substitute of the gasoline. And then when we use the Clostridium, it produces the butyl alcohol, acetone and carbon dioxide, which is largely used in the pharmaceutical industries. Now, plant system also, this is the metabolic pathways. You can say several important metabolites is produced as caffeic acid. This is used as a stimulant. It is largely available in the tea or coffee. Then it produces ethylene also. This is used in the ripening process. Then limonene and mannitol, that is also another product that form in the plant system. And in the biofuel production, as I pointed out, starch or glucose that can be produced by the different type of this is different type of hydrocarbons, that alkanes or alkenes, the formation is there, and this is the different other products that formation nowadays. This has largely exploited, particularly by using microalgae, and microalgae used as a source of biodiesel. So in conclusion, I want to tell that biochemistry deals with different biomolecules, like we have carbohydrate, we have proteins, we have lipids, we have this nucleic acid, and that mainly the carbohydrate and lipid they use as a source of energy, and protein used as a bodybuilding material, carbon also used as a bodybuilding material, a part of carbon, and nucleic acid mostly used as a genetic material for the formation of different proteins, which is very much involved in the metabolic pathways. There are several metabolic pathways that we have, as for example, endo and myro pathway, where glucose is converted to pyruvic acid and pass through the TCA cycle, it produces carbon dioxide. Other metabolic pathways also has been discussed, through which we can get the different type of products, both primary metabolites and secondary metabolites. Thank you very much.